Hello everyone, welcome back for another session of regeneration biology class, and we will continue with the liver regeneration with some knowledge. We will try to gain more knowledge on the mechanistic insights of liver regeneration. In the last class we learned how different cell types gets regenerated, how the cell cycle regulation takes place, and how various signaling molecules contributes. And today we will try to understand a little bit more elaborate at its mechanistic level, and what are the different ways of injury, and what are the ways of damage a liver gets and how it is responding. So the scenario that we can explain in this following manner: the liver possesses a distinctive capacity for regeneration within our body, and it's one of the complex organ that is capable of remarkable regenerative capacity. This we have already seen it. Under normal circumstances, the liver cells, they replicate themselves and maintain the liver function. This also we have seen it, because the hepatocytes must replicate for retaining their normal function, because many hepatocytes gets damaged and they will be defunct because of the detoxification process that takes place in the body. Usually the replication of healthy hepatocytes is sufficient for the regeneration for acute liver injuries, normally, depending upon the extent of acuteness. When I said detoxification, we should understand it need not necessarily come from food or atmosphere. It can also happen the metabolic toxins that your body is producing. Say, your body produces lots of ammonia, and your body produces lots of carbon dioxide, and your liver is powerful in converting that into urea, which is eventually filtered by the kidney. So the ornithine cycle which takes place in the liver, which is a detoxification process. So ammonia is dangerous, carbon dioxide is dangerous, urea is also dangerous, but it is less dangerous. That doesn't mean that it is not a toxin. In advanced phase of chronic liver damage, a large number of hepatocytes die, and the hepatocyte replication is blocked because of the extent of damage. That time the hepatocytes are no more able to manage the damage. So the liver regeneration has more complex mechanisms, such as transdifferentiation, that is conversion of cell type A to cell type B. So that also we saw in the previous class. And they also, when the damage is very severe, the hepatic progenitor cells comes into picture. Their distribution is there. That location also we saw there besides the canals of Herring. But they come into picture only when the damage is severe. Inadequate liver regeneration causes severe chronic liver disease, and often the patient dies. Even in animal models, if the liver is failing to regenerate, the animal will die within 2 weeks. There is no way the animal can survive if the liver is not recovering. So this is a schematic distribution of various cells in the liver. So those who are interested can read this citation, which is a review, very interesting review, which talks about, so these pictures are taken from that review. And this is a liver. As you can see here, it is a multilobular structure. And if you take a magnified portion, or take a portion that is under a microscope, if you take a cross section, it looks somewhat like this. You can see the blue colored one is the vein. Usually the veins are represented blue in color, and the hepatocytes are light brown in color, and these portal veins are usually distributed on an average. It is around in a hexagon manner, as you can see here, and there are dedicated sinusoid regions are there, which is going all the way up to the central vein. The portal vein goes and joins into the central vein, and also there are dedicated sinusoid regions, and which are the place where the signaling events takes place after injury or a repair. And also you can see this green colored ducts which is coming towards the central vein, and this is the bile duct. the green color portion, and it also have got a long structure called bile canaliculus, which is the extension of the bile duct, which eventually becomes a hepatic duct, which is releasing its content into the boundary of small intestine and stomach and the small intestine. It joins, it normally releases the bile acids. And if you take a magnified portion of this further, you can see this is the portal vein and hepatic artery, which supplies the fresh blood, and you can also the distribution of Kupffer cells, and you can also see a cholangiocytes, which is a dedicated cell that is embedded into the hepatocytes. The brown colored ones are the hepatocytes. You can also see Kupffer cells. They are basically the macrophage, which basically doing the phagocytic function. And you can also see the stellate cells, which are all important for various liver functions and also for the signaling events. So this is the scenario. This is how the cells are distributed. So to study the regeneration, you need to have a injury model. Any regeneration study require a proper injury model, and the injury model is selected in such a way that it mimics the natural damage a organ can get it. So one of the easiest and well studied model is the amputation model, that is also known as PHX model, that a surgical procedure is performed to remove a specific section of the liver without causing direct harm to the liver cells, or the organ is functioning as it is, a portion is being removed. And there is an advantage of doing this because you can damage consistently. Say if you are doing by some other method, say you put a toxin, and it depends, the effect of that toxin depends on the ability of this animal to deal with the toxin, and it won't be same for all. Whereas amputation is concerned, it is going to be fixed. If you amputate a given quantity of tissue or remove a given quantity of tissue across different animals, the damage is going to be more or less same. However, there are other methods like this. Chemically induced liver injury models also exist, because we know the persistent damage of the liver that takes place due to various chemicals. Chemically induced liver injury models include the administration of diethyl nitrosamine and carbon tetrachloride or ethanol. We know ethanol is one of the major cause of liver cirrhosis all over the world, because the ethanol creates an intermediate called acetaldehyde, which is very dangerous, very toxic, and it can damage the liver in no time. Even in this injury, chemically induced model, there are two types or two subcategories are there. Acute liver injury model. In acute liver injury, it often occurs when a high dosage of toxin, let it be any of them, let it be diethyl nitrosamine or carbon tetrachloride or alcohol. You give a high dose in minimum time, less amount of time. That is one way, acute liver injury model. And there is chronic injury model of the liver, where the chronic injury is induced with a sustained or mild or very moderate levels of the toxin at a frequent interval. It is something like that of a person getting addicted to alcohol. No one start drinking huge quantities of alcohol. People start slowly and keep on increasing the dosage. Very small quantity, your body may be able to deal with it, although toxin is a toxin, but you keep increasing the dose. And something similar, it's been done in a chronic liver injury model. However, in a laboratory condition, chronic injury method takes a lot of time to standardize, because if you are doing in a rodent model, we don't have the luxury of running the show for 6 months or 1 year, because the mice will grow old. So hence, many a times the PHX method, that is the amputation, or the acute liver injury model is being used. However, chronic liver injury model also has been done, because that is the most prevalent in the case of human. For instance, long-term excess alcohol consumption or chronic viral hepatitis sometimes happens, like say for example, hepatitis C virus. Once it entered your body, it never leaves. It is always there. All you can do is you have to support your liver as long as you want to live. There is no way you can get rid of hepatitis C virus. you may have heard about in news, some celebrities have got infected. So if it normally spreads through the blood transfusion, once it entered your body cannot clear, which is same with many other viral diseases also. But some chronic viral diseases, what happens, that viral hepatitis always keep on damaging your liver. Such scenario, to study such scenario, you need to have chronic liver injury models as well. So this is a picture that shows, it's taken from the same review. You can see the chronic liver injury can happen due to type 2 diabetes also. So obesity is one of the major cause. When you are eating a lot, your body deposit those carbohydrate or fat in your body with a cause which is damage to your liver, because liver has got fat depositing cells and your body do not want to lose a single nutrient that is put into your body, even though it is damage to the body. So you end up to start with getting a fatty liver disease. And fatty liver means your body has got disproportionate amount of fat that is deposited, and it can manifest in the form of some liver pain, etc. But body try to repair, like if you reduce your food intake, this fat will be metabolized. If you do exercise, etc., it will be metabolized and the liver will reduce the fat load in the body. However, if it has switched into a cirrhosis stage, then the liver has to turn into the regenerative mechanism, and hence the problem can come from various situations. And normally the fatty liver or the obesity related liver damage happens because of insulin resistance, which is nothing but type 2 diabetes, and excessive alcohol consumption, and the hepatitis causing viruses. Different viruses are there, hepatitis A, B, C, like that. C I gave as an example. Another example can be some tumor that can come in the liver, and it can start consuming a lot of nutrients that is coming in the liver, and many times doctors will do a surgical approach to get rid of the tumor if there is no secondary spreading, etc. That also something similar to that of a traumatic liver damage. And of course, in a natural environment, if two animals are fighting, if horn of one animal entered into the belly of another animal, its liver can get damaged. So these are all something which occurs naturally, and no animal can afford to lose its liver because it has to survive its body. So these are all traumatic brain injury. And then lot of chemicals which you take, like even simple paracetamol, what you are eating for fever, the maximum your body can tolerate paracetamol in a day is 8 gram. Thankfully, every paracetamol is only 500 milligrams. If you take 10 paracetamol in a day, it is only 5 gram. But more than 8 gram in a day can cause liver damage, because the paracetamol also need to be detoxified. Like that, many other tablets and chemicals which you are eating daily, no, even if it is not studied for the liver toxicity, some drugs can cause damage to your liver. So it is always good to stay away from the drugs unless it is essential for the living or the survival of this animal. Drugs should be life-saving drugs rather than some people take a painkiller just and then keep playing the game or keep damaging your body further. So basically, if pain is tolerable, better not to get into a painkiller or any other drug. Now let us see what are the growth factors that can influence the regeneration of liver after a liver resection. It can be, like I told you, it can be any damage. Hepatocyte growth factor and epidermal growth factor. These are all the two predominant growth factors. Hepatocyte growth factor, also known as HGF in the short form, they are secreted by the hepatic macrophages such as Kupffer cells. They secrete HGF and they mediate the hepatocyte proliferation through its receptor c-Met. c-Met is a receptor that is essential for a given hepatocyte to respond to the HGF. It activates downstream effectors such as extracellular signaling regulated kinase, also known as ERK1 and ERK2. And another gene, downstream gene that is activated because of HGF binding onto the c-Met is protein kinase B, and also known as AKT. Protein kinase B is another name is AKT, and Then comes signal transducer and activator of transcription known as STAT3. So ERK1 and 2, AKT and STAT3. Remember, this pathway can be turned on by other mechanisms also, but HGF binding onto the c-Met receptor indeed can trigger these mechanism, and these are all pro-mitotic. That means it will trigger the cell into a proliferative phase and driving the hepatocyte proliferation and its survival. Just the proliferation is not good enough. Unwanted proliferation, your body will make it die by apoptosis. So this is also you should keep in mind, that whenever there is a proliferation, not that it will stay forever. Your body is waiting for killing all type of cells that is formed inadvertently. But here these mechanisms also ensure that they don't die by preventing apoptosis. Epidermal growth factor EGF. EGF is another factor like HGF. EGF is through its binding site is onto a receptor called EGFR, means epidermal growth factor receptor. It initiates the hepatocyte's proliferation signaling pathway and it promotes the hepatocyte proliferation and its survival. So both HGF and EGF contributes to the hepatocyte proliferation and its survival. In the PHX model, that is the amputation model which we discussed, after blood flow was restored, soon after cutting there will be a blood clot and the blood flow will restore back. HGF and EGF are rapidly activated to promote hepatocyte division and proliferation. So it is one of the prime response is through HGF and EGF induction. And the other signaling that is induced are Wnt proteins. In the previous class also we discussed about one of the Wnt and how Wnt is contributing, Wnt is playing a major role in the proliferative event, and Wnt proteins and signals. So regeneration after hepatic injury relies heavily on Wnt beta-catenin signaling. So normally I can quickly explain, the beta-catenin is produced in every cell. Every cell needs beta-catenin for its survival. It's part of the cell-cell communication. Cytoskeletal, like tubulin actin filaments are connected with alpha-catenin. Alpha-catenin is linked with the beta-catenin. Beta-catenin is linked with integrins. So this is needed for every living cell need to have this beta-catenin. However, the excess beta-catenin is degraded by a process called destruction complex. We will not go into the details of destruction complex, but understand destruction happens inevitably to the beta-catenin so that it never get a chance to go to the nucleus. It will be degraded in the cytoplasm. However, when the Wnt binds to its receptor called frizzled, what happens? This destruction complex is no more able to function because Wnt binding to the frizzled receptor, it will take away one of the pivotal players of this destruction complex. Then it's like you have a four-legged table. One table I broke it. Then that table will not stand. It will three-footed table or a chair cannot stand. So all four legs are important. So one leg will be snatched by Wnt frizzled complex. Hence the destruction of beta-catenin will not take place effectively. This stabilized beta-catenin will now go to the nucleus, and with the help of another collaborator called a TCF, they will bind onto the DNA targets and turn on transcription. So this is what happens. Following PHX, the Wnt beta-catenin axis is activated to facilitate quick liver regeneration by facilitating hepatocytes to enter the G1 phase. All hepatocytes are in G0 phase. They will now enter the G1 phase, promoting the DNA synthesis in a cell. Additionally, by controlling the downstream effector genes such as cyclin D1 and c-Myc, these are two downstream expressing genes of Wnt beta-catenin signaling. And this cyclin D1 and c-Myc, they will favor the proliferation of a cell. The Wnt signaling interacts with other signaling events such as NOC and HGF, because Wnt signaling can influence the production of ligands of delta NOC signaling like jagged, DLL, those kind of genes are turned on by Wnt signaling. So they can influence the delta NOC signaling and also HGF. hepatocyte growth factor production, which we discussed some time ago, forming a complex regulatory network. Then comes the presence of cytokines. Cytokines are what? Secreted molecules that can influence a target cell. They are normally of two types, broad categories, pro-inflammatory cytokine and anti-inflammatory cytokine. So they do either of this function. In general, the cytokine is a chemical, mainly proteins, protein chemical or peptides that can drive a cell to perform something. And this is the simple meaning of cytokine. Interleukins are classic examples such as IL-6, interleukin 6, IL-33. They exert important roles in liver regeneration. After hepatic resection, that means damage to the liver and acute liver injury through a single dose of a toxic chemical, etc., IL-6 contributes to a lot to the liver regeneration. Because IL-6 is a very predominant pro-inflammatory cytokine. During COVID, you may have seen many of the people who died, their lungs, their bloodstream had exceptional levels of IL-6, which makes their body, of course, it is because produced from your own body's immune system, but beyond certain limit of IL-6, it will be too difficult for your body to handle. But here in liver regeneration, the IL-6 has to be induced to make the environment pro-inflammatory. After hepatic resection, acute liver injury, IL-6 contributes to a lot to the liver regeneration, and IL-6 mainly secreted by the caseins or Kupffer cells activates the Janus kinase, JAK, and signal transducer and activator of transcription, Janus kinase and STAT3. They are called JAK-STAT pathway. So JAK binds to the STAT, and the STAT is normally staying in the cytoplasm but never gets phosphorylated. Because of this binding, it gets phosphorylated and goes to the nucleus and acts like a powerful transcription factor, STAT3. Signaling pathway, JAK-STAT signaling pathway, after binding to its receptor, IL-6R. Interleukin 6 has got its own receptor, so it will activate only those cells which has got IL-6 receptor. So IL-6 is secreted, IL-6 bind down to those cells which has the IL-6 receptor, and it will turn on JAK and STAT3 pathway. And IL-6 JAK STAT3 signaling promotes the G1 to S transition. If this is G0 to G1 is done by HGF and EGF, now it has to move from G1 to S. Then only cell cycle will proceed. So this JAK-STAT pathway has the power to convert the cell from G1 to S phase, that is DNA synthesis phase. This is mainly done by upregulating the cyclin D1 expression. If there are no cyclin D1 expression, then it will not go into the G1 to S phase. So STAT3 plays a major role. STAT3 activation induces the upregulation of several anti-apoptotic genes such as MCL1 and BCL2. So these are very powerful anti-apoptotic genes. Even if you have some potential of many cancer cells use this strategy because cancer cells don't want to die. So it will turn on. A healthy cell should not have these MCL1 and BCL2 levels. If these genes are high, even a mad cell or a cancerous cell will not die. So you don't want that to happen. But in this case, it is okay because the STAT3 makes sure that the BCL2 levels are gone high, MCL levels are gone high, thereby enhancing the anti-apoptotic ability of hepatocytes in the injury environment, because there any new cell is welcome because the liver badly want to gain the bulk. IL-33, interleukin 33, induces intestinal mucosal cells because it's coming into the bloodstream, mucosal cells to release more of the serotonin, because 95% of the serotonin in your body is produced by your intestine, not in the brain. Only 1 to 5% of the serotonin is in your brain, rest are produced by your intestine, and it will release more of the serotonin into the portal bloodstream, and it activates a HTR2A P30S6K in hepatocytes, which is also helpful in the proliferation. So now comes the bile acids. In the previous class also we discussed in the parabiosis experiment, the bile acids are one of the components that influencing the liver. So bile acids, the hepatocytes create bile acids which are then discharged into the biliary duct and the intestine. It gets eventually into the intestine before
being absorbed again. So it goes into a circle because it is released and immediately it will be released once its job is it is absorbed, once its job is done. The bile acids are returned to the liver via enterohepatic circulation. There is a unique circulation called enterohepatic circulation, which is releasing and absorbing because bile acids are mainly like a detergent. What is the use of detergent in cleaning your laundry? Same job. It acts like a emulsifier. Like lipids and liquid water will get into an emulsion with the help of bile acid. Bile acids levels dramatically increased immediately after PHX, that means partial hepatectomy. It is called PHX in rodents and humans. And bile acids accumulate, the bile color, the coloration of the pigment becomes yellow in case of jaundice, and if your liver is not functioning, you look yellow in color. Bile acids accumulate something called farinosoid X receptor, also known as FXR, and then another G protein coupled receptor known as GPBAR1, and it's also referred to as it has another name, that is TGR5. These are all some names of the genes and the receptors, and they're playing essential functions in regulating the bile acid balance and preventing the liver damage. Once damage is there, you don't want further damage to happen. FXR activation stimulates gene expression in bile acid synthesis, detoxification, and transport. So this helps to reduce liver damage caused by bile acids and supports the liver regeneration. Furthermore, we should know that the bile acids modulate the activity of various growth and regeneration related pathways. For instance, the Wnt beta-catenin signaling. Moreover, the FXR and TGR5 just now we discussed signaling have been demonstrated not only to promote hepatocyte proliferation but also protect hepatocytes from apoptosis. Production is one thing, protecting is another thing. So both they do. So it produces and protects, thereby facilitating liver regeneration following injury by partial hepatectomy method. You can see here in this picture signaling pathways, what we have seen, different methods. Whatever we have seen is mentioned here, that EGFR receptor and MET receptor, and it binds, and PA3K, AKT, mTOR pathways turned on, and the proliferation kick starts. And you also have here CCR5 receptor, CCL5 binds, and you have got CCR1 receptor. It all turns on FOXO3A signaling pathway, which in turn activates the macrophage, and then also it will eventually produce a lot of hepatocyte growth factor. And you also have the IL-33, which I discussed just now about activating the intestinal mucosa to secrete serotonin, which eventually reaches the liver, and it turns on HTR2A and P70S6K pathway, which also helps in the liver regeneration. And there is something very interesting, that is hypoxia inducible factor, which normally formed because of the supply to the blood supply to the liver is cut. The HIF1A and NRF2, these will stimulate the production of some unique, and also the level of cholesterol, because cholesterol is something which is produced in your liver, and if the cholesterol level is fluctuating, that means the level is low, that also is an indication of your liver malfunctioning. They also can stimulate starting from the cholesterol level, and this it will trigger the HIF and NRF2, and it will trigger the activation of liver regeneration. So in general, we can see liver regeneration, chronic liver damage, which happens by a normal proliferation of the hepatocytes, or which is normally can happen through the dedifferentiation, and also it can be dedicated stem cells can come into picture, and they can contribute to the Wnt signaling and the jagged production, and the NOC signaling get activated, and the NICD go to the nucleus, and it can turn on a bunch of genes like DNMT, YAP, TED, SOX9, and also hepatic nuclear factor. They all make the liver cell able to regenerate. We will study about a new subject in the next class. Thank you.